So this is under the nervous system YouTube links. There are 3 videos that sort of give you a good visual for what the nerve actually looks like. This one here, though, how the synapse works and the nerve impulse, you might want to come back to again when you have, it's definitely the nerve impulse, when you are trying to explain it in the middle of that assignment that you're going to do on electrochemical impulses, just to see it one more time. But I want you to see it for the first time, do some work on it, come back and view it again. Okay, so let's take a look at this guy first.
Trying to introduce the study of the brain to a bunch of students. I said if understanding everything you needed to know about the brain is a mile, I asked the students how far had we walked in this mile. I got answers, 3/4 of a mile, half a mile, someone said 1/4 mile, and I said I think about 3 inches. This is unconventional science. There's just this huge gap in our knowledge about how brains work, largely because we have no idea what they actually are made up of at the finest level. When it comes to the nervous system, there are a large number of diseases where the only real sign that there's something wrong is the outward manifestation of the disease. A person is acting crazy, or they don't seem to learn very well, or their movements are disordered in some way. But if you look at their brain, most of the techniques we have, there's nothing to see. You've got to see the wires. You just have to see them, and you have to see where they come from, where they go, what they connect with, and be able to map that out in enough detail that ultimately you will be able to render the information that goes into a brain to see the wiring diagram at the resolution of every single synaptic vesicle in every synapse. Enough resolution, we think, to see everything in a wiring diagram. I'm a lab head of a group of people who work together. It's kind of a shifting group of people, but it's difficult work and it requires many kinds of experts. So there are lots of people working together on this project.
What we're trying to understand is how cells in the brain communicate with each other. I have to look at the entire pipeline from the beginning, where we start with a mouse, make a sample, process the tissue, cut sections, and then ultimately begin imaging them. A slice is like you're cutting a loaf of bread, except you're trying to slice very thin pieces of tissue. So for a millimeter depth, you get about 33,000 sections.
Each section of the brain is next to the section that was sliced just before it, and the slice right after it is the next frame. And if you play these frames in sequence, you see the brain not over time, but over space, as if you're looking deeper and deeper into the brain. The big objects you see that appear and then slowly go out of appearance, those are nerve cells that are being cut through. And coming off of these cells are these large light-colored objects, which are the dendrites. Every little object in here is a little wire. To keep track of these cells from one section to another, what you'd like to do is kind of color them in. So every one of these colors have no meaning other than to keep track of each object from section to section. What this allows us to do is to generate a wiring diagram based on this. We now know everything about these wires here, and from that we can generate all the connections of every cell in that area. Information forces you into this uncomfortable position where you have to kind of say, okay, I don't get it, but I know that the real world is more complicated than the way I'm thinking about it. I feel it's a very long road, and we've just started. That's my view.
Just in this picture alone, before I actually click on the whole thing, because I want to be able to see the thumbnail, you've got nerve cells that it's one big continuous cell, but it's got this weird projections that kind of their cytoplasm is pushed out so that their cell membranes are in these weird little shapes. So this is one big giant cell, And this little section here that comes out, this is called the cell body. This is where you'd find the nucleus. This one little section that comes out here is called a dendrite. So each of these little weird things are dendrites. And those are going to be connections that are going to receive information in the form of neurotransmitters. They're going to receive chemicals from other cells that have just dumped them into a gap. That gap is called a synapse. So anytime you hear about synapses, we're talking about a gap between cells where you're going to dump these chemicals that are going to talk between the two cells. So the dendrite will receive the information, and it's going to start something called a voltage gates. They're going to be flipped. You're actually going to have electricity traveling along in the form of ions flipping across the membrane all the way down until you get to the other end, where you're also going to see these connectors. From the cell body, this little stuff that's wrapped in pink here, this whole little extension all the way to the end where it has its weird little, looks like little like gecko feet, that's called the axon, that whole thing. And this is the direction to the right, is the direction that you'll have the actual electricity flow, or the voltage will flow along that way. So the message always goes from the cell body end to the axon end. So received by dendrites, goes to the axon and keeps going. Right? At the end of the axon, there are little neurotransmitters, little chemicals that can be dumped into the next synapse, the next gap, and then could trigger or could inhibit, actually, depends what type of neurotransmitter you have, and it could excite the dendrites of the next guy and then decide whether or not it's going to take that message in and continue on with it. All right. So that's what the cells kind of look like, and now I want you to see what's happening in the gaps between the cells.
In the last module, we took first steps towards understanding the electrical properties of individual neurons. We learned how electrical forces and diffusion give rise to membrane potentials, and we learned how cells can generate and propagate signals called action potentials, or spikes, along the membrane. Understanding the properties of the neuronal membrane is essential, but understanding just these properties isn't sufficient to give us insight into collective behavior of the billions of connected neurons in our brains. Luckily for us, we can approach neuroscience at many different scales and levels of analysis, and we don't have to confront the full complexity of everything all at once. That's what we'll be exploring throughout the rest of this course as we slowly go from our understanding of single molecules, such as ion channels, to the electrical behavior of neurons, to their collective behavior in small circuits, and finally onto how they become organized in large functional regions of the brain. Let's start simple though. Since we've examined one neuron, a logical next question is, how did two neurons connect with one another? We'll first examine some basic cellular anatomy of neurons. So far, we haven't made too much of the fact that the majority of neurons are polarized cells. That is, they have one portion of the cell for receiving inputs and another portion for sending outputs. The parts of the cells that are specialized for receiving inputs from other cells are called dendrites. The word dendrite comes from the Greek word dendron, meaning tree, and as you can see, the dendrites have a branching, tree-like structure. A signal received by a dendrite is passed to the cell body. If there is sufficient depolarization of the cell body membrane to initiate an action potential, then an action potential is sent down the axon. The axon then carries the propagating action potential to another neuron. So what actually happens at the boundary between two neurons, between the axon of one neuron and the dendrite of another? This interface is called a synapse. There are two general types of synapses: electrical synapses and chemical synapses. Electrical synapses are less common in our own nervous systems, but they're simpler to think about, so let's start with them. Electrical synapses are basically pores between two cells that allow ions to pass through. They allow the passage of the electrical signal through to a neighboring cell without much fuss. It's not so different than just combining two cells into one larger cell. There are lots of reasons that nature might need synapses like this from time to time. They're fast, and they allow cells to couple together with a high degree of synchronicity. But most neurons are connected together by a much more complicated structure called a chemical synapse. In a chemical synapse, rather than simply passing along an electrical signal from one cell to another, the action potential travels to the end of the axon and causes a chemical to be released into a very small space between the two neurons called the synaptic cleft. This chemical is taken up by the downstream neuron on the other side of the cleft. This chemical signal can cause the downstream neuron to depolarize its membrane, converting the chemical signal back into an electrical one. Or it can have other effects on the cell. This chemical step is slower than transmission across an electrical synapse, but it opens up an enormously diverse repertoire of different and more complex kinds of signaling. And synaptic function plays a critical role in computations performed by neurons. We'll spend the rest of this unit exploring the inner workings of chemical synapses, on our way to beginning to look at how networks of interconnected neurons give rise to behavior.
Okay, good enough for that one. Just to give you an idea of the gap of the synapse, where you have a neurotransmitter produced by the one axon gets dumped into the gap, which triggers a response on the next one. Creepy little mouth and ears on this particular version of plasticine stuff, but that's okay. So just to get the idea, that's how they get the chemical message across. Sometimes that chemical message, those neurotransmitters, are telling the next one to fire, so they're excitatory. Sometimes they're telling the next one, don't you dare fire. You need to stay quiet. I want this guy to fire instead. It just depends what the message is and what direction you want the message to go. You don't want one neuron to be able to trigger a response all over the place. You want it to have a direct pathway so that the information gets where it needs to go in the brain. If you do have an occasion where all of a sudden all these things are firing all the time, that would be a problem. That's where epileptic seizures come from, when there's this firestorm in the brain, or electrical storm, I should say, in the brain, of too much information, and the next thing you know, the body's overwhelmed and you're having a seizure. Not good. So you want to be able to direct pathways. So sometimes you want to say, go yes in this direction, but please don't go that way. And they want the message to be very directed. So that's one of the reasons why we'll have excitatory and inhibitory, but you'll learn about that when you do your little assignment shortly. Okay. Now you were seeing those plus and minus flipping along the membrane there. That's the idea of membrane potential gets set up. Remember in our first unit we talked about the sodium-potassium pump? In one direction you're shooting potassium and then it opens to allow sodium to go the other direction. And it takes energy to do it, to change the shape of it, to be able to pump. So we're going to be back to talking about the sodium-potassium pump. I'm going to show you here how it actually ends up working. And well, in your little assignment you're going to do it. I'm not going to directly teach it, but that's what this last little video is going to show you. Okay, it's going to talk to you about how you get that flipping of membrane potential. And I'll pause as we go if I need to.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge.
So let me just reiterate what he just said there. So you've got this pump that's able to, for every couple of potassium that you throw inside, you're going to lose 3 sodium, 2 to 3. So you're going to pull potassium in at the same time that you're pushing sodium out. There are other ion channels that once the potassium is inside, let me go back a frame. Once the potassium is inside, there are other channels there that will allow the potassium to leak out, but there's not really any structure that allows the sodium to leak in. So you're going to maintain, you've got lots of positives on the outside, and the only thing that you can do is get even more positive on the outside if these ions leave. Okay. It's the difference in charge between those two areas that make one positive and one negative. So in order to get a nerve to fire, this pump has to reset the nerve so that we get this positive charge on the outside and a negative charge on the inside. The only way for the sodium to get back inside is if you trigger something called a sodium gate. So in other words, it's like this little, it's like a membrane, a protein, but imagine it's got a cap on the top of it. And that cap is blocked for now. But if you can trigger that first little guy to open the gate, then the sodium will just come rushing in through facilitated diffusion. So you pumped it out using energy, but when the time is right, you're going to open the sodium gate and it'll flow in. And what's going to be neat is you've got sodium gates all the way along the membrane. And once the first sodium gate gets triggered, so will the next one, so will the next one. So it's like dominoes fall. And then you'll be able to suck back in all of that sodium that was on the outside simply by facilitated diffusion. You pumped it out a little bit of energy, but it's going to move back in through facilitated diffusion when the time is right. The way that we know the time is right is if at the very beginning, remember that dendrite at the very beginning, that thing that looks like a tree, the very beginning on the edges of the cell body, they have receptors there that can receive information from a neurotransmitter from a previous axon of a previous nerve, right? So, neuron, I should say. So it dumps that chemical message. So if it is enough of a message with a neurotransmitter to flip the first sodium gate, then you'll see the message will travel across the entire thing right down to the end and will release neurotransmitters at the other end to try to trigger the next guy as well. Okay? So it's about creating this voltage difference. Voltage is just difference in charges, right? It's the same thing within a battery. A battery only works as long as it's able to separate charges. You get positives building up on the one electrode, and you get negatives building up on the other electrode. In this case, the electrode is the membrane itself. So you've got it on one side versus the other, and you have the ability to prevent the movement of at least the sodium until you flip that gate. You'll see that as we go through here.
Exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside of the cell.
So now that you've had the signal where you had the sodium flow in through the gated channels, then you had it also trigger potassium to leave. So now I've got more potassium on the outside, more sodium on the inside, and I'm going to have to pump to reset and get it back to having the sodium on the outside and the potassium on the inside. Okay, and again, that potassium is free to flow. It'll get pumped in, but it can flow back out. But the main thing is getting the sodium to the outside. Okay, this is also why, if you're thinking about what sodium and potassium are, this is why electrolytes are important in your body. These are electrolytes that we're using for this type of thing. You'll see the same kind of sodium gates and all sorts of stuff happening when you're talking about muscle fibers firing as well. So we're going to use electrolytes to deal with muscle contractions, and we're going to use electrolytes here to deal with our actual membrane potential happening along there. Okay, so the idea of them flipping and having that negative to positive flip here, that's the action potential as it moves along. So every time you see action potential as a word, you're thinking, okay, that's the message itself, the electrical message being sent along. Different neurons are going to have different voltages. You'll have some really high voltage neurons and very low voltage neurons. So some of them will have a much bigger charge difference is what we're talking about. Way more positives on the outside compared to the inside will get you a bigger voltage. That's the charge separation that we're dealing with here. Okay. So let's run through then, again, the anatomy of nerve cells. One of the things that you need to know is that an actual nerve cell, the conductive cell, the one that actually has all those sodium gates that we just saw, the one that sends the actual message, that's called a neuron or a neuron. It just depends on Oxford versus Webster, I suppose, which dictionary you're going with for how to pronounce it. But same word, it's a neuron. When we're talking about nerves, usually we're actually talking about nerve tissue. So this is where, unfortunately, the language gets kind of blurred, because you can say one neuron is a nerve, which is how we're generally talking about it. But really, if it's a nerve bundle, you've got a lot more going on within that nerve tissue. Tissue is more than one cell type working together. You're going to have a lot more going on. And in fact, if you were to actually look at what is the majority of your brain or any of your nervous system, it's not actually a neuron that is your massive amount. It's actually these glial cells or neuroglial cells. And the word glial is just the Greek word for glue. It's the stuff holding everything together. But it's more than just a connective type of tissue. These glial cells are going to do these four things here. They're going to provide structural support, so keep the right neurons in the right place. They're going to help feed and give energy, supply oxygen, all that kind of stuff, nutrients to these overworking neurons. They're going to make sure that not every neuron touches every other neuron, because we're going to want the signal to go in one direction, not in all directions. So if I can insulate and stop a neuron from being too close to the neuron beside it, it'll never dump neurotransmitters in its direction. So it keeps things directional for us. And there are some types of glial cells. There's lots of different ones, but some of them are actually up there just making sure that if any virus or bacteria does cross the blood-brain barrier, that they're there to help clean it up, or they're there to help take care of any of these dead neurons that are causing, they just need to be cleaned up. They're at the end of their life, or they've been damaged or something, and they need to be cleaned up. Just like we have immune cells in other parts of our body taking care of other types of cells, some glial cells have this immune function to them as well, the cleaning up and tidying up function. Right? One of the neat things about glial cells is that they are able to talk to each other. So they have a way that they can signal and say, come this direction, go that direction, we need help over here, whatever it is. They can talk to each other using calcium ions. So there's a communication system there. And one of the things that I think is very cool, if you can get a chance to take some kind of anything to do with embryogenesis, that just means how the embryo develops in the womb. If you can do any kind of developmental biology, even if it's just like the extra course that you take in your first or second year, take it. It gets very interesting when you're looking at how do we go from being sperm and egg fused together to being this complex 100 trillion cell masterpiece, really. I'm not pointing at myself, I'm pointing at all of us. Like the way that our bodies fit together and work is really quite amazing. How do different tissues actually develop? At what stage does a cell know, okay, right now I'm a stem cell, embryonic stem cell, but now I know I need to become nerve tissue, or I need to become muscle tissue, or I'm a heart cell. Like when does that message happen, and how does it know? One of the things that they have discovered is that these glial cells, one of the first nerve cells that you're going to get would be a glial cell, specialized, and the glial cells can actually guide and direct the main neurons to know where to grow within the body. That way you've got the connections in your fingers that are supposed to be in your fingers. So you have your touch sensors, you've got your temperature sensors and your pressure sensors and your pain sensors that are in your fingertips, all that kind of stuff. We had to know, how do you get the nerve to grow so that they go to the right point? And the glial cells are part of that process. They help to guide and tell the neurons where to go. So it's kind of interesting that they can talk. It's kind of like a shepherd leading the sheep as you're developing. So if you get a chance to take an embryogenesis class, it won't be called that. It'll be called developmental biology or something like that. It's very interesting stuff. Okay. What you're looking at here in the yellow is one complete neuron. And what you're looking at attached are the different types of glial cells that can help to aid. So the astrocytes and the oligodendrocytes, you don't need to know that. You can just call them glial cells, but they will have different roles doing different things, either helping feeding, helping with immunity, helping to guide, Or what this guy is doing here with the oligodendrocyte is it is actually wrapping around and producing something called a myelin sheath. Myelin is a type of protein. And when it puts this protein onto this axon here, it's going to speed up the transmission of that particular nerve. Okay? Some of the glial cells are also going to help to secrete that cerebrospinal fluid. That's going to be important for feeding, bathing, cushioning, all sorts of stuff. And they're also going to get rid of extra neurotransmitters. Neurotransmitters, like acetylcholine and whatnot, are norepinephrine, are these little chemicals that get dumped into the synapse that trigger the next neuron. But you don't want them just sitting there all the time. You want to be able to clean them up so that you get a clear message the next time you dump neurotransmitters. You don't want them endlessly just in a pile there, triggering things you don't want them to trigger. So once you dump neurotransmitters, you need to clean them up. And the glial cells will be responsible for making sure that happens really well. Right? And then again, what they'll do is wrap around, just like you see with the blue here, and they'll make that myelin sheath, which will make your nerve transmission on that particular axon much, much, much faster. The answer to why it's much faster is a little more complicated. There's a video there as well to help you, but I'm not going to show it to you yet. I want you to do the actual information run through how all these things are happening with the textbook first when you do that little assignment, then watch it at the appropriate time to help you kind of get a better idea of what's going on. Otherwise, it's just going to wash over you right now, probably like those first two videos just did. Okay. In the central nervous system, you have glial cells doing slightly different things than out in the peripheral nervous system. Remember that PNS means peripheral. The Schwann cells, it's obviously named after a dude, the Schwann cells out there are, when they myelinate those axons, they're actually also there to help repair neurons. So when you break a nerve, cut a nerve somewhere out in the peripheral, nerves do grow back. I know you've probably heard that once you have a spinal injury, that's it, you're done, which is more likely true in the spinal cord, but it's not very true out in the peripheral. It takes a long time, but it will grow back, hopefully in the right direction at the right time, hopefully sooner than later. So for example, I've cut my finger really, really badly back in February of 2006. I was cutting a cardboard box. I lived on campus when I was teaching in India, and I heard the school bell go, and I had to run to my grade 9 class. And it's like, 2-minute run, 4-minute walk to my class. But the bell went. I thought, oh, shoot, I've only got 5 minutes. So I went a little faster with this big carving knife to cut a box down, cardboard box, and I slipped right at the end, and the big knife went right through my finger, like right down to the knuckle, right to the bone. So I had this little flap, whatever, and I'm bleeding and everything. I ran. Luckily, there's a little hospital on campus, and also wonderful, I had a lab assistant, like someone who was hired to set up labs for me. So I was like, Ruby, please just set up the lab. I'll be back in a second. I just got to go to the hospital. Went down a little farther, got there, wrapped, passed out, got a few stitches, went back to class, and finished my day. It was quite the day. The only reason I'm telling you about this weird story is that I cut the nerve. When I cut my finger, I cut the nerve. Right? And for the longest time, and even now, it's still a bit weird, I had no sensation, I couldn't feel anything being touched there. So heat was gone, pressure sensor was gone, pain sensor gone, because I cut that particular type of nerve right down to that section. So you can feel everything on this side, but right there's a dead spot. It's kind of weird. So for the longest time, if I was holding something hot, I just used my left hand because it didn't feel so bad when you're holding a hot coffee because I can't feel how hot it is. It was great. It started to come back though. So 2006 is when I cut it. And I'd say probably in the last 2 or 3 years, it's not completely back. There's still some weird dead spots in between, but I'm starting to feel again. I'm feeling temperature. I'm feeling all the things I should be feeling with that nerve there. Your peripheral system, because of these Schwann cells and because of these, the myelinated stuff, I'm able to repair those ones a lot better. Now, your central nervous system, the gray matter, this is white matter, the gray matter doesn't have this potential to have this quicker repair mechanism. So if you were to break a neuron in the central nervous system, its growth would be incredibly slow, if at all. That's why spinal injuries will take a very long time to heal, if ever. Most likely will never heal, because they don't have this added advantage of the myelin sheath created by these Schwann cells. Right? The other problem, and I mentioned it in the last lesson, is that you have these astrocytes. I'll show you that one again. These guys here, whose job it is to repair and do all sorts of stuff like that in there. And if an astrocyte instead of helping to repair a neuron that doesn't have the myelin sheath, if it gets in there and instead forms scar tissue, then you'll have literally a physical barrier between this neuron and the next one. You're not going to be able to send transmissions because of that physical barrier. Right? So the scar tissue doesn't have the same conductive properties as the regular neurons that should be there. That's a problem. If I can just grow a new neuron, or I can take this neuron and grow new connections, maybe the cell beside this one was damaged, but this one can reach out and can take the place and replace that guy and say, hey, I'm here to help. Give me the message, I'll send it. Then that's a possibility as well. And again, those Schwann cells are going to help to promote that kind of repair and to myelinate. So we speed up transmissions by myelinating. That's what makes it white matter, because it actually looks white, not blue like this picture. And then you're speeding up the neuron repair. Right? Around every single axon, so again, look at this picture down here, you do have this neurolemma. It's a very thin membrane, but it's in your PNS, not in the CNS. And again, that's the part that's helping to regenerate those axons. So it's in the peripheral nervous system, not the central nervous system. Okay. Again, white matter, nerve cells that have the myelin, they look white. Gray matter, you can see they don't have it. They don't have that protein wrap around it, no neurolemma, so they cannot regenerate properly. Multiple sclerosis, you've all heard of MS before. Perhaps you even have members of family or friends who have been affected by MS. It's actually quite a terrible degenerative disease as it's progressing. It's quite sad because there's nothing wrong with. It's not like you couldn't speak before or couldn't walk before. It's degenerative. It's as you go through, what ends up happening is the myelin sheath gets destroyed. You can have hardening. You can have scarring happening. That's what the sclerosis part is. It's a hardening. And then what ends up happening is you don't send messages as quick as you did before because the myelin's gone, and you don't send messages as smoothly and as necessarily where they need to go. And that's what leads to double vision, where you can't speak properly, or jerky movements with your limbs. You're not coordinated in the same way because you need so many different parts of your brain talking to one another in order to make smooth motion, in order to be able to produce the type of speech. So the words are in your head, but you can't get them out using your tongue and your jaw and your vocal cords in order to produce the correct type of sound to make any sense. So what you're saying is what is in your brain, but you can't get it out. So it's quite a tough thing to walk through if multiple sclerosis is in your future. So tough thing to go through. But they're leaps and bounds all the time on what they're doing with research and how they're trying to fix this and what can they do about it. And so hopefully one day this won't be a thing that we talk about. Maybe we'll talk about it the way we talk about polio. Like, you guys don't probably even know what polio is because it's been eradicated by vaccinations. But it's another type of thing like this. Hopefully one day we'll be able to just cure multiple sclerosis. So neurons then, if those are all glial cells, all the supportive role glial cells, help support, help speed up things, help repair, all that kind of stuff. Then the neurons themselves are the conductive cells. These are the ones that actually have the voltage gates and have the electricity flow through with the voltage. There are different types. You can have a sensory neuron, and sensory neuron is going to take information from a sensory cell, something like the cones and rods in your eyeball that are sensitive to light, and then they will transmit that information to the optical nerve, and then that heads up into the occipital part of the back of your brain there. And then it'll go to the temporal, and the temporal and occipital will figure out what the heck it is you're looking at. So the sensory neurons are going to send information to the central nervous system. The motor neurons are going to receive information from the central nervous system. And in terms of your testing, I'm going to be asking you just about the structure of a motor neuron. I don't need you to memorize both. This is basically the same idea anyway. But what we're going to do is talk about the fact you got a cell body. You have these dendrites sticking off of here. Then you're going to have this axons all the way down. Now, they've cut the picture because these can actually be quite long. They may as well have just left it short. But so then you will have this axon going all the way down, and then it'll have the axon terminal or terminus will be at the other end, and that can either directly affect something like a muscle cell or something like that, or it can be giving the message to another nerve to continue the transmission. The point being that sensory they are afferent with an A, afferent on its way to the central nervous system. Motor neurons are efferent, E, efferent, coming from the central nervous system. And both of these are parts of in this particular case, are parts of the peripheral nervous system. So again, talking about the parts here, you got your dendrites, which are these parts sticking off over here. Then you've got your cell body or soma. I don't care which way around you call it, just know what it is. So the cell body part where the nucleus would be. Then you've got your axon and your axon terminals. And if you're lucky and you're a myelinated one, you'll have the myelin sheath around the outside. It's a Schwann cell, is the name of the cell. The protein it produces is myelin. That forms that neurolemma. In between, those Schwann cells are not completely joined all the way down. You'll have these little gaps in between, and those are called the node of Ranvier, or node of Ranvier, depending on how French you are. Node of Ranvier. So these are what's going to appear to happen, although this isn't technically what happens, but what it looks like is it's going to look like the signal doesn't have to go through the whole length of... So remember we had the sodium gates, right? Flipping, 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 flipping. Because of the width of the cell at this particular point, because this extra wide membrane is sitting here, you're going to have much faster nerve transmissions. It's just one of the things that if you have a wider nerve, you're going to have faster transmission. That, again, there's a video that'll show you the details of why. I'm not going to try to explain that right now. But you're going to have slower transmission in here and in the gaps, and you're going to have very fast transmission here. The reason you're going to need to have these little gaps in between is because as the signal goes across this thicker section, it starts off strong and then gets weaker, 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 and then it goes strong again in this little skinny section. So this is the action potential that I'm talking about. It spikes again, so I got a big strong signal, and then it's weak, weak, weak all the way down, spikes again, weak, weak, weak all the way down. And so you've got these nodes of Ranvier in between because you're trying to boost the signal enough to get it so it's very fast across the little fat bits, but it'll peter out if it's all just fat. It'll be too slow of a message. So fast, slow, fast, slow, fast, slow. And what ends up looking like, it looks like it just jumps between the nodes, but it is actually passing along the entire signal until you get to the axon terminus. Terminus just means end, or terminal means the end. That's why we call it a bus terminal. It's where all the buses end up. And then this is where you'll take those neurotransmitters and dump them into the synapse gap to trigger the dendrite of the next guy. One of your test questions is going to be asking you about how do you speed up the nerve transmission, speed up or slow down. If you get a fatter axon, you're going to, in other words, it's wider, bigger diameter, you're going to be much faster. And that's what the myelin sheath is going to achieve for us, a nice fat membrane. So that's why myelinated is faster. So either just on its own, it's a wide axon, and then on top of that, you add myelin sheath. Now you've got a super fast one. Again, we've talked about this already, but the neuron feedback loop, if you've got a sensory neuron, okay, it's going to get, look down here, signal pathway. You're going to get some kind of a stimulus, heat, sound, light, whatever it is, one of our senses. It's going to hit a receptor cell. That receptor cell triggers the sensory neuron, sends the information to something called an interneuron. An interneuron would be things you can see up above here, mostly located in the brain and the spinal cord for the vast majority of them. That makes some kind of a decision and says, this is how I need you to act, and sends the go do this message to a motor neuron, which hits an effector cell, like a muscle cell or whatever, and then that's how you get your response. Please memorize the signal pathway. Stimulus stimulates a receptor cell, tells the sensory neuron the information, sends it to the interneuron to make a decision. Motor neuron gets the message back to the effector cell, and then you have your response. Again, sensory neurons are also called afferent, and then your interneurons are called association because they are the link between. They associate the motor with the sensory. And then the motors are the efferent because they're going from the CNS out to the PNS. Okay, types of sensory neurons that can be connected to different types of receptors, like photoreceptors in the eye, your smell receptors, your chemoreceptors in your nose, pick up chemicals, thermoreceptors in the skin, etc., etc., etc. Okay. I'll let you read through to see the rest of those ones on your own. The simplest neural circuit that we have is called the reflex arc. The reflex arc doesn't even need to worry about the brain. It's going to be sensory nerve or neuron to the interneurons that are located in the spinal cord. and they will immediately make their own decision without having to ask the brain and send the information back to the muscle. So let's say, again, you touch something hot, your thermoreceptors in your skin are going to sense the heat. Then that sends information to the sensory neuron to your interneuron in the spinal cord, key that it's spinal cord. It decides that's too hot, we need to move the hand away, sends the information to the muscles of the arm and the hand to contract, pulling that arm away so that you're no longer burning. And while the message is already on its way to move the hand, your brain is starting to figure out the fact that it's hot. And it's simply a distance thing. It takes time for these messages to travel. And so you want the shortest path possible when you're in danger to try to get the message and the response as quickly as you possibly can. It's much faster to get to the spinal cord than it is to go all the way up to the brain, have a conscious thought, gee, that's real hot, I should probably move my hand. Okay, let's move the hand, and then move it. By then you're already burnt. You already have crispy fingers by then, right? So this is the basic idea. They're going to be involuntary, you're not controlling them, and they're often unconscious. Those are two different things. You can have an involuntary response, but be aware of it. This is involuntary, and it's already happened before you become aware that it's happened, right? So you move hand, then you yell and say, ow. And here again is another picture depicting that kind of an idea. And you're going to have, again, the receptor comes into the dorsal side, interneuron is in the actual spinal cord, that's the gray matter there, sends information back out the white matter, out the myelinated one, back to the motor neuron, to the nerve, through the nerve cell to the effector cell, boom, I'm done. And I've already had my response before I even know it. Okay? So we will be doing this reflex arc activity shortly. And it's in the old text. You guys are going to grab, if you're in the room, old texts are on the side. But for this particular thing, both of these things I've actually just put into the booklet. For the other stuff that we're going to do later, there is so when you go to, then you're going to do this assignment here. This assignment also needs the textbook. If you're in the room, you're using the physical book. If you are at home, this has been posted on the classroom. Just go to content and you'll find it there under either under textbook or look for this assignment and you'll see the textbook pages have been there. Okay? That's all I have to say for now. That's the end of this little lesson.